My name is Mats Ljungman. I am the co-director of the Center for RNA Biomedicine here at Michigan, and I'm delighted to moderate this seminar today. We have two new faculty members that's been recruited to Michigan, and we're going to start with John Prensner, who is an assistant professor in the Department of Pediatrics and in biological chemistry. So John got his MD and PhD from University of Michigan. He did a residency in general pediatrics at Boston Children's Hospital. He did a fellowship at the Dana-Farber after that, and finally a postdoc at the Broad Institute. And then we recruited him here to Michigan last summer. And John has carved out a very interesting research area that he calls the dark genome that he's going to tell us more about today. And if you have questions, please type them in to the Q&A sign in the bottom of your screen. So John's going to talk about functions of non-canonical open reading frames in cancer. So please go ahead, John.
Thanks so much, Mats, for that introduction. I'm just going to share a screen now and give me a thumbs up or a thumbs down. I think we should be all set to go.
Yeah, it looks good.
Great. Well, so as Mats mentioned, I'm a relatively newly minted assistant professor here at the University of Michigan, and I'm a pediatrician. So, I have to keep the atmosphere in the room kind of kid friendly. So we like monsters and monster movies in pediatrics. And so I always like to think about how do monsters play a role or not in the research that we do. And so here you can see actually we've got a plethora of monsters. And so I'm very excited to share them with you today. So I don't have any disclosures that are relevant. I have one licensed diagnostic to Decipher Biosciences. It's unrelated to this talk. And I think, so the topic at hand that we think about is this area of genome and cancer science called non-canonical open reading frames. And what are those and why are we excited about them? And so I think when most of us, or at least when I was, I guess, growing up and receiving my education, you learn about a conventional protein coding gene and what they are in mammalian biology and human cancer biology. And most protein coding genes have a coding sequence called a CDS, flanked by leader sequences such as the 5' untranslated region and the 3' untranslated region, which are felt to be mainly regulatory at the RNA level or promoters. And there are maybe 20,000 or so genes like this in the human genome. But what we've learned really in the past maybe 10 to 15 years is that there's this other side of the coin, that there are these 20,000 protein coding genes, and yet there seem to be huge numbers, some numbers of thousands of additional things in the genomes as well of these cells. And these are open reading frames that appear to be translated, and they can be located in long non-coding RNAs. So these may be either be bifunctional RNA slash translated RNAs, or they could just be misannotated link RNAs. They also could be in the confines of an existing annotated gene located perhaps in the leader sequences, such as an upstream open reading frame, or in the downstream sequences. And the numbers and prevalences of these are highly speculative at this point. But what we can say is that they tend to be relatively poorly conserved. They tend to be quite small. And if you do traditional mass spectrometry, you don't find them so well. But you do find them quite well if you look at the immunopeptidome or ribosome profiling, which is a technique to use sequencing to map where ribosomes are moving across an RNA to incorporate amino acids and tRNAs. And so these have really opened the door to studying these types of open reading frames in human disease and how they are now enriching our knowledge of human cancer. And so for us, what we've been thinking about for some time is not really the question of do these exist? We know they exist, and we've known for quite some time they do, but more, why should we care? And is this something that we really should be directing our attention to for patient discovery and for clinical medicine purposes, or is this really something that maybe is less exciting from that perspective. And so we think about these open reading frames, in different possible ways. How much is this stochastic in the cells? I guess we're not entirely sure at this point. How many of them are regulatory, but maybe not primarily protein coding, is another big question of discussion. And how many of them actually make what we might call functional microprotein, which would be a kind of real protein in the way that you might think of a protein. That is even a further question. And what we've been doing is trying to really address this case of functional proteins. And what we do is take these large numbers of open reading frames that are known to exist and found in cells, and use experimental techniques in the lab to understand which ones are translated and which ones actually might be functional microproteins in cancer that we should sort of direct our attentions to. And so the hypothesis here is that there are going to be these dark genome microproteins from these non-canonical open reading frames that are going to give us new disease insights and maybe, in fact, new biomarkers and generate new therapeutic hypotheses kind of going forward in the future. So the task for the rest of our time here together is to talk a little bit about the functional genomics that we do to think about these in cancer. We'll talk about how microproteins are genetic vulnerabilities in cancer, and we'll zero in on some exciting recent work. And we'll talk about how we can actually shift gears now and think about you in your labs, and how do we bring this now to you and have it not be so hard to access and sort of mysterious, but how do we actually get these open reading frames into your hands, into your labs, so that you can study them. So the first story I'm going to tell you about briefly is just work done a number of years ago at this point during the postdoctoral fellowship I did at the Broad Institute in Dana-Farber. And what we did at this point in time was say, we know these open reading frames exist. How would one study their biology at a large scale to be able to sort of give us confidence or sort of reduce our confidence in whether or not we should study these further in the context of cancer? And so we designed a number of functional genomic screens. Now in 2024, they seem rather primitive, but when we were doing this, 7, 8 years ago at this point, it was quite exciting at that time. And what we did was design CRISPR knockout screens for a large number of these open reading frames and apply those to cancer cell lines and think about how these open reading frames may be actually required for cancer cell life and viability. We also then developed a large number of cDNA plasmids encoding the open reading frame and looked to see whether that was sufficient to induce gene expression changes in the cancer cells. And what we found is that actually a very large number of these cDNA products could actually make a detectable protein in the context of that exogenous cDNA ORF expression. This is not necessarily saying in a real human cell they really make a protein. It's just saying in the context of an experimental system of exogenous expression they could. We further found that when we exogenously expressed them, almost a third of them could actually make a gene expression change in cancer cells. And this we thought was due to the translation of the open reading frame as opposed to the transcription, because if we made just subtle point mutations to change methionines to alanines, that was typically 2 methionines per ORF on average, we would completely lose the ability then to induce this gene expression change. And then when we turn to the exogenous human genome sites, the cancer genomes, when we knocked out those endogenous sites, over 10% of them in this study actually seemed to have a role in the maintenance of cancer cell viability. And so for us, this was quite exciting. I think it sort of suggested that maybe we should invest more time in this area as compared to discourage us from investing that time. And we went about thinking about how do we now really bring this into a realm of cancer biology that's much more concrete, much more sort of palpable and potentially important for patients. And so for this, we turned to actually a multidisciplinary team and a great collaboration. So this was work done by my group with Ian Yanuzzi and Rakesh Morgan-San, as well as Sebastian van Heesch at the Prinses Maximus Center in the Netherlands, and Damon and Jorge. And it just came out maybe last week in Molecular Cell, so I'm happy to share it with you here today. And so what we turn to in this case is a devastating childhood brain cancer called medulloblastoma. This is, in fact, the most common childhood brain cancer that is malignant. Even so, it's still relatively rare. Only about 500 patients in the United States a year, or children per year, are diagnosed with this. It's most common in infants and toddlers, which makes it all the more sort of devastating because often we're treating very young infants with this disease. And it's a highly aggressive disease. It can spread throughout the brain and spine. It can even spread in rare cases outside of the brain and spine, although that's quite uncommon. And as such, we really treat it with very aggressive treatments. We use surgery, we use chemotherapy, and we try to use radiation, except in very young infants as well. And what we've learned, though, over the years as a sort of pediatric oncology community is that it's not one disease. It's actually a spectrum of diseases. And these are quite molecularly defined. There are 4 basic subgroups of this type of disease. There are additional sort of more nuanced subgroups as well that we won't get into today. But suffice it to say that 2 of the subtypes have typically good prognoses. So those we expect patients to typically survive their cancer and do well with treatment. But in other subtypes, particularly ones driven by the MYC oncogene, really patients have quite poor outcomes, and these are highly aggressive cancers. And so we wondered whether or not the transcription factor biology that really is a hallmark of some of these negative, poor prognostic cases really might be due to translational changes that are occurring at the level of maybe promoter or leader sequences, independent of how these oncogenes activate gene transcription. So the model is that as they are activating gene transcription, maybe they're also having some influence, whether indirect or direct, on RNA translation. And so what we did was profile medulloblastoma patient tissues with RiboSeq and RNA-seq and perform CRISPR screens on the things we found from that. And the first high-level analysis was simply quite compellingly to show that RNA translation is specifically perturbed when you have this MYC transcription factor biology, and it's unique beyond just gene expression changes. So it's quite straightforward to say that a transcription factor like MYC, when it's high in these cancer samples, will cause a unique transcriptional signature. That's been well, I think, described. What's more interesting is when you integrate RiboSeq, you can then do this thing called translational efficiency, which is really thinking about how many ribosomes are distributed per open reading frame. How actively are these transcripts being translated in a post-transcriptional way? And that is also quite perturbed across cancer subtypes. So we do think that there's strong mechanisms driving this in this disease. And if you just look at the overall translational efficiencies, you'll see that in the MYC-driven cases, it tends to be compellingly increased compared to the MYC-low cases. So this was interesting to us, and we can sort of parlay this information then to think about translational regulation of some of these non-canonical reading frames, where we could easily detect some thousands of them in these medulloblastoma patient samples. We could use these information then to think about studying them in the context of disease and disease subtypes by designing specific CRISPR screens that, in this case, really focused on the question of whether or not the translational control of these upstream open reading frames was significant. So we could design CRISPR guides that would knock out an upstream open reading frame compared to knocking out an annotated coding sequence, and ask the question of how often does this one have some function in the cancer that's unique and not present there. And so we specifically looked for ones that cause the cancer cells to die when knocking out their adjacent region did not cause them to die. And we could follow this up actually with even higher precision CRISPR work, where we could actually saturate the genomic turf. And here what you're looking at is each dot is a guide RNA and done in 3 different cell lines. And when you specifically have guide RNAs for this upstream open reading frame, the cancer cells will die. But really, if you have anything else in that genomic vicinity, they don't really seem to mind it one way or another. So we were able to do this actually in about 7 cancer cell lines, quite a large number in total. And we stratified those cancer cell lines then based upon their molecular biology to ask the disease question of how does this relate to MYC? And how does this inform us about aggressive patients and the treatment that they receive in the clinic? And we could find a small number of cases where actually certain upstream open reading frames were increased in their essential requirement in these cancer cells compared to the cells that did not have MYC in them. Among these, perhaps the most compelling was this example that we found in the ASNSD1 gene. This ASNSD1 gene is actually relatively poorly characterized. It produces a 600 and some amino acid protein. Itself is not a cancer gene, not known in cancer, not really known to be regulated in cancer either, to my knowledge. But what we could find is actually when MYC is present, it dramatically upregulates the translation of this upstream open reading frame at the mass spectrometry level as well. And we could model this in vitro where we could express, in this case, MYC-N and show that it also goes up at the protein level. Furthermore, we could go back to the patient tissues and show, yes, this upstream open reading frame is selectively correlated with the MYC or MYC-N transcription factor abundance in patient tissues. Now, all of this is only true for the upstream open reading frame. None of this is true for this ASNSD1 coding region. And that was particularly interesting for us because this UORF is also, we think, a genetic target now in this disease that can be pursued. So what you're looking at here is each dot is a guide RNA where we're knocking out this genomic site. And the pink and purple colors, those are MYC-driven medulloblastoma cell lines. And the grayish colors, those are non-MYC-driven cancer cell lines. And what you can see is that this knockout of the ASNSD1 gene doesn't tend to do too much, but knockout of the UORF has quite a strong effect, particularly in these MYC-driven lines that gets attenuated as you get closer to the C-terminus end. That's actually typical for these screens, so that's an expected result. So what was this thing doing in medulloblastoma? I don't have time today to take you through all the data. We did a number of work on this, but basically what we could find is that it creates a microprotein that then binds and interacts with what we call this pre-folded-like complex. This is a hexameric protein complex, 6 proteins, that we believe has a role in chaperone or protein folding at the post-transcriptional level. This is now at the amino acid and proteomic level. It is, in fact, related to this pre-folding complex, but is indeed more mysterious because it actually only retains 2 of the 6 subunits between these two. And so really the roles of this have never, to my knowledge, been studied in a human cancer. And so we did a number of experiments to show that this UORF interacted with this complex and correlated in patient tumors with this complex quite highly. I don't have time to show you that, but what I can show you quite interestingly is the downstream functional consequences of what we think it's doing. So when we knock out this UORF and the prefolding complex in these medulloblastoma cancer cell lines, we observe a highly active signature of proteins that get regulated by them. This is only true at the mass spec level. You do not see this at the RNA-seq level, so that confirms a post-transcriptional mechanism here. And particularly for cancer relevance, it appears to cluster on proteins that are involved in cell cycle. This is a highly important cancer signature. So we could show this again in patient samples as well, where I know this is a little busy, but we could take patient samples that had RNA-seq and mass spectrometry, and in essence, use the logic to say, if this complex regulates the proteome and not the transcriptome, then when this complex is highly active, you will have a widened difference between the proteome and the transcriptome because the complex would be imparting more regulation. And that's, in fact, exactly what we did see. I know the slide is a little busy, but we call this dyssynchrony between the proteome and the RNA. And this dyssynchrony goes up as this UORF goes up. And it actually correlates with the targets of the UORF that we can find by knockout in vitro. So this, again, then correlates in patient samples with cell cycle and translational regulation genes. And we think this is now maybe a new mechanism of how the cancer cells are trying to regulate themselves as they sort of advance their aggressiveness and disease phenotype. So the model here is that we could now take a human disease driven by transcription factor, MYC in this case, show not just transcriptional changes, but also translational changes of ORFs, use functional genomics to identify unique microprotein ORFs of interest that are coming from the upstream open reading frame areas, and do some downstream molecular biology on one of our best candidates. Now, in the last 2 or 3 minutes here, I just want to talk about how do we bring this now to you. It's fine if we can do this in our group here, but wouldn't it be lovely if more people could do it and there'd be more research and more dissemination of knowledge across the scientific community? But it's really hard because finding these ORFs, figuring out what they are, and thinking about them is not trivial. And for those of you who've thought about ribosome profiling experiments or non-canonical open reading frames, know that there's no real standard way necessarily to analyze the data. It's not like RNA-seq, where there's a relatively well-established pipeline. You can take these data and you can look at it multiple different ways. You can find all the messy, nitty-gritty stuff in all its glory, or you can take the data and you can look much more sort of clear-cut answers. And it's not easy to know what to do with it. And so what we've been doing now internationally with a group of RiboSeq researchers is trying to identify best practices for labs that want to do this work. And you can see one citation here that I direct you to for details. But what we can see in basic is high differences in the performance of different ways to analyze these data, that you can use the same data set on biological replicates and get a 10-fold different result in what you're identifying and different confidence levels. We can sort of use that information to identify ways to look at the data that we believe might be high stringency or low stringency, either of which could be appropriate depending on your research question, but it's important to know what you want to get when you're going into the analyses. So in a high stringency approach, you're looking at, say, thousands of UORFs. In low stringency approaches, you're going to get 10,000s across the board. And then the question arises, what is ground truth? And that is really, I think, a hard question to answer. So we've turned to our international community. This is an old slide, and so I apologize. This is now updated with more groups, but there's a number of us who now work together in this consortium to try to think about how to standardize these open reading frames for all of you, the RNA biology and the research community that may be interested in them. And so we're asking these questions about, how many are there and how do we think about them in terms of proteins and how do we figure out what to put in a database for all of you to use. Where we're at right now is working together with GenCode and other arbiters like UniProt and HGNC to think about how do we really name these consistently, how do we annotate them in the genome consistently. And in fact, we've been able to produce a first catalog of consensus non-canonical ORFs with GenCode, and you can download it from their website there. And I encourage you all to think about this if you're looking into this type of research for your own lab. So in summary, at this point, we currently think that there are many of these non-canonical open reading frames in the cancer genome. We think that numerous non-canonical open reading frames, many of them are going to be genetic dependencies in cancer, maybe disease targets, and ultimately that these non-canonical open reading frames may encode microproteins that are going to give us this new layer of insight to cancer that helps to open up maybe new biological discoveries. So, sorry, I'm going the wrong way. So the last thing just to comment on is in the clinic, why should we care? And I think part of the reason for that is that these are going to potentially offer a new avenue to biomarkers, disease tests, and potentially therapies based upon maybe immuno-oncology, where we're finding potentially immunostimulatory roles for some of these non-canonical reading frames, or maybe drug targets or molecular glues that come from them. So ultimately, what we're driving for is dissecting the RNA biology of these translated ORFs in order to advance the clinical vistas for our patients. And so with that, I'd just like to thank first the patients and advocates and foundations that really drive much work in pediatric oncology. You can see a number of them here who've supported our work, and really we're grateful for all of that, as well as the folks who really helped drive this project, Ian Yanuzi and Rakesh Murugan-Sand, who were working with me, as well as Sebastian von Heesch and Damon Hoffman in the Princess Maxima Center. And with that, I'm happy to take any questions, and thank you for your patience.
Thank you, John. That was awesome. That's really good stuff. And congratulations on your new paper. And also commend you on organizing this international consortium of looking for these, going over this dark genome in a broad sense. So that's really cool. Yeah, if you have questions, please type them in. I can start with one. So you focused on cancer, but do you think these ORFs play a role in lots of other diseases?
Yeah, so we absolutely think that they're important in other diseases. In fact, probably the most mature scientific community for this area is in human genetics. And in November, we had the honor of presenting actually a seminar, a symposium at the American Society of Human Genetics, where a number of human geneticists talked about all the research they want to do about finding mutations in these things, right, for human genetic diseases. And so that is a very high area of interest, I think, for the research community. And there are a couple of different ways to go about that I think I'm not a human geneticist, but I think there's a number of folks in that community who are going about that.
Mm-hmm. Great. We got another question here from actually our next speaker. She's asking, is the dark genome only full of microproteins? Are there many standard size, non-canonical, unannotated ORFs as well? If not, why? You often find large ORFs in alternative ORFs in any gene locus, no?
So some of this is, I think, wording and nomenclature, and it's a little bit thorny to get around some of these things. To take a step back, there is no size definition for a microprotein. So often we'll say less than 100 amino acids. That's really a personal choice. These can certainly be up to 150 or 200 amino acids long. It becomes much less common. Why? Because when the human genome was being annotated, they used, at that time, the state-of-the-art computational systems to identify novel protein coding sequences in the human genome with a threshold of they had to be greater than 100 amino acids. And so the Human Genome Project did a pretty good job of finding things that were greater than 100 amino acids, but it was still a little fuzzy if you were kind of close to that mark. So 150 amino acids, not too far from that. So there is no size threshold for a microprotein, but the majority of what we find are quite small because those were the ones that were not systematically investigated during the Human Genome Project. I don't know if that fully answers the question, but I think that's maybe the most succinct way to put it.
Okay, great. We have a few more here. So Niels Walters asking, I might have missed it, but what mechanism leads to the overexpression of these UORFs in cancer?
So Niels, you didn't miss it. We don't know.
No.
So there are two aspects to this. One is expression at the RNA level. The other is translation at the RNA translation level, I guess. And at the level of RNA translation, we don't know. And that, I think, is an area of ongoing investigation. Is this direct or is this indirect? Are there specific ribosome subunit factors? Are there specific eukaryotic initiation factors that couple with these? There's been some literature of excitement around a molecule called DAP5 and its ability to identify UORFs and leader sequences, and then in that case, sometimes selectively regulate them, perhaps driving them down in that case. But in the main, we don't know. And right now, we just observe the phenomenon that they do get selectively regulated at the level of translation.
Great. Fabrice Leclerc is asking, any specific feature of these non-canonical ORFs in terms of codon usage?
Yeah, there are some codon specificity so specific usage. It tends to be arginines and glycines. Many of these upstream open reading frames exist in 5' leader sequences. In general, 5' leader sequences are more GC rich due to DNA methylation, et cetera, that regulates promoters in that way. And in general, Gs and Cs lend themselves to arginines and glycines and a few others, maybe prolines. And so those do tend to be enriched in these on the whole. Other than that, not really, I guess, that I've seen.
Okay, great. Last question here from Eric Malikos. Do you have any thoughts as to why disproportionate amounts end up on MHC1? Is it true in vivo, or could it be because cells in vitro are not being co-cultures with CD8 T cells?
Yeah, great question. I'll take the second one first. This is true in vivo. So this is true in human patient samples. It's true in mouse studies. It's not a cell culture thing. The disproportionate amounts. So the going thought is that many of these open reading frames, if they produce a protein product, produce an unstable one. And the going thought is that the number of stable microproteins, the way that we talked about today with ASNSD1, that's probably a comparatively small. What percentage, I don't know exactly, but it's not the majority. So the majority of these likely make unstable products that then get degraded by a proteasome mechanism. And proteasomes funnel peptides then into the MHC class 1 system. By contrast, MHC class 2 is not coupled with the proteome and is not enriched in these non-canonical open reading frames.
Okay, great. So that's all the questions we have time for. Thank you so much, John. It's great talk.
My pleasure. Thanks so much for having me.
Yeah, thank you for doing this. So our next speaker is Michelle Hastings. She's a professor in pharmacology and in medicinal chemistry here at Michigan. She got her PhD from Marquette University in Milwaukee, and then did a postdoc with Adrian Craner at Cold Spring Harbor, where she worked out some of the mechanisms for SMA, and actually took part in some of the early steps in producing the first approved ASO treatment for SMN, and I guess any kind of ASO used in the clinic. It's called Spinraza. And so after that, she became an assistant professor at Rosalind Franklin University in Chicago, where she rose to the level of full professor. And then in May, we were fortunate to be able to recruit her here to Michigan. And here she serves many roles. One is the director of the mRNA Therapeutics initiative. So Michelle's going to talk today about splice-switching antisense oligos for treatment of disease. Go ahead, Michelle.
Thank you, Mats. It's great to be here. I'm going to this one.
Yeah,
great. So I'm gonna. Are you good with the screen? Perfect. All right, just a few disclosures here to report. Before I get started, I do want to put in a plug for our 2024 RNA Symposium that's happening Friday, March 8th. We have some wonderful keynote speakers and a whole day actually of great talks and activities planned, with a particular keynote, Drew Weissman, who just won the Nobel Prize. We're really excited about that. I also want to announce a new section that's coming to the RNA Journal. And that is a section on RNA therapeutics. And I'm going to be the editor of that special section. And I really encourage you and hope that you all submit your papers, research articles, short reports, communications, lots more to come about what this section is going to feature. So I encourage you to look into that in the near future. I also want to talk, just let you guys know that there's a new society called the Society for RNA Therapeutics that is just starting to launch, and you can become a charter member for free for a limited time. And so another exciting thing that's happening in the RNA therapeutic landscape. So now talk about my lab and some of the work that we're doing in this exact space. So my lab is a RNA biology and therapeutic discovery lab is how I like to describe it. And here we are, most of us in our current state. I want to just say happy birthday to Jacob Sikowski. I'm not going to be talking about his work today, but he's a great graduate student. So happy birthday. We study RNA mechanisms, basic mechanisms, splicing microRNA exosomes, as well as therapeutics that target RNA or use RNA approaches. We're pretty agnostic in terms of diseases, really more interested in ways to modulate and manipulate RNA to treat disease. And then we like to collaborate. I think that's the key to success, especially when you're really working across using therapeutics across many different disease and human condition platforms. And so I'm excited to present here at the RNA Collaborative Network and and would love to hear from any of you if you're interested in some of the tools and approaches that we're using. So I'm going to give a little bit of background about antisense oligonucleotides and why they make really nice therapeutics, some of the features that have really launched them into the forefront as a therapeutic platform. And then I'll give an example of one of the projects that we've been working on in the lab, and a little bit of new work that we have in that regard. So antisense oligonucleotides, and their really most simplest definition, is just a short nucleic acid that functions by complementary base pairing to a target RNA. And we use them to modify gene expression, and that can either affect the protein or an RNA. Antisense oligonucleotides have been around, and the concept has been around for a long time. And what has really made them more drug-like is modifications in chemistry. And so this is just a few. This is become a very large field, the chemistry of nucleic acids. These are some types of them shown here and some of the features. So the backbone modifications have increased nuclease stability and nuclease resistance and stability. Sugar modifications have improved the specificity and the affinity, and other modifications have helped with that as well as delivery. And so there's just a lot of features about that you can modify to improve their drug-like qualities. There's a number of antisense oligonucleotides now that have been FDA approved for the treatment of disease. And there's really two types of these short antisense oligonucleotides, and we kind of call them steric blocking or occupancy only mechanisms, and then RNase H targeting gapmers. And there's been, again, several, and these approvals have come pretty quickly in the last few years, and it's many, many more that are in clinical trials, and I expect to see a lot more drugs from these types of antisense oligonucleotides in the near future. I really want to just stress again the difference, because there's sometimes a lot of confusion about the different types of ASOs, and it's important because, first of all, they do different things, and second of all, they behave differently, and they have different features in terms of drugs. So steric blocking oligonucleotides have a fully modified backbone typically, and then they have modifications that make them that they have modified sugar rings that make them resistant to degradation. So they just bind and they block the RNA. And this can lead to a number of different effects depending on where you target them to. RNase H targeting are similar in the wings to a steric blocking, but then they have a center core of about 10 nucleotides that are unmodified in their sugar ring, and this makes them more DNA-like. And then when they base pair to RNA, they're degraded by RNase H and degraded. So it's really a way to downregulate gene expression. And this makes them a little less stable, and so they're not as long-lasting as steric blocking. And that DNA-like backbone also gives them oftentimes some features in terms of their the reaction in vivo that is different than the steric blocking oligos. In terms of mechanism of action, as I said, these RNase H targeting or gapmers are generally used to lower RNA or protein levels. So it's a pretty, like siRNAs, for example, they kind of do the same type of thing. Whereas steric blocking are a little more, you can be a little more creative with. They've been used to inhibit translation and also to activate translation, depending on where they base pair, whether they can block inhibitory or enhancing sequences. They can block microRNAs. They can block polyadenylation in the nucleus. And what we use them for primarily is through splice switching. And this is what all the drugs to date so far have been developed to modulate pre-mRNA splicing. But really, they can be used at any level of protein maturation or stability. And there's a lot of activity and interesting work coming out about how you can modulate gene expression in different ways with these for therapeutic value. Delivery is also another huge field around here. So I have one really simple slide. The the splice-switching oligonucleotides to date have all been delivered just as a naked RNA. Some of the gapmers are conjugates, so that helps them to get in by the conjugate binds to a cell surface receptor, and that can give cell type specificity. Lipid nanoparticles are another way that's being investigated, or polymers and things like this. So there's a lot of activity around that, but they are able to get taken up in vivo, and I'll have some more slides about that also. And then the administration routes today, these are some of the administration routes for the approved drugs through sub-Q, intravenous, intramuscular, intravitreous, and intrathecal for the CNS. So the oligos are not, so far, none of them are can pass the blood-brain barrier. So the ones for the central nervous system are delivered through an intrathecal or lumbar puncture. Some other really great features about ASOs. This is one of my favorite papers of last year. It was really great. Looked at single cell map of antisense oligonucleotide activity in the brain. So after either an ICV injection into mice or an intrathecal injection into non-human primates, and they did single cell nucleus sequencing and looked for where these were gapmers, so they were an oligo that was knocking down gene expression. They looked in the different cell types to see where the oligo was getting. And so you can see here, mice after 1 week after a dosing, and anything to the left of this line shows the knockdown. So you can see in every cell type that they looked at, they were getting pretty good, almost every cell down, really good knockdown. And this is really useful to know when you're thinking about what cell type you want to target for a particular disease that might involve a very specific cell type. And this is a time frame. So over 12 weeks after a single injection, you can see you start to lose some activity here, but it lasts after one injection for quite a while. And then in non-human primates, similarly, after 13 weeks, 4 IT doses, you get really good knockdown in all the cell types that were looked at. So that is really, I think, speaks really to some of the pharmacokinetic properties and dynamics of this. In terms of pharmacokinetics, this is another a really important feature of oligonucleotides. And this was a study done with nusinersen, which is the FDA-approved treatment for spinal muscular atrophy, or Spinraza, the first splice-switching oligonucleotide in the CNS. And this was given to NHPs. And what you can see after a single injection here in the spinal cord in particular, I point out that after 52 weeks, you still have high activity of that oligonucleotide. So the ASO concentration is still very high, with a half-life about 97 days. So that, and you can see also it stays circulating in the CSF and in the plasma, although that is very quickly lost and then stabilized. Sort of another thing about antisense oligonucleotides that is kind of a unique feature of a drug is that because their mechanism of action is inherent with them, you can design them to base pair where you want, and so you know what the mechanism is. They can be developed very quickly, and so this was, I think, a real game changer for ASOs and personalized therapies when melicin, an antisense oligonucleotide for a very rare mutation in Batten disease, was developed by Tim Yu at Boston Children's. Mila was a child with a disease diagnosed in 2017, and an ASO was very quickly developed in Tim's lab, and then administered within a year to Mila, and that really changed a lot of the landscape for this type of oligonucleotide in the case of not just having drugs for more common diseases, but also for rare diseases. And the FDA issued this guidelines for N of 1 antisense oligonucleotide therapies, very specifically giving us the kind of a roadmap for how to do this for other diseases. And since then, we've have companies, private companies, as well as foundations and other organizations that have really started up to enable these types of N of few studies, and making us really see the potential in the future for personalized genomic medicine. And Lorem has now begun treating 6 nano-rare patients. They have a lot of applicants for their developmental pipeline. And clearly showing the need for more of this and the interest, and I think a real unmet need for the future. So now I'll just see where I'm at with time. Good. Okay, so I want to get a little bit more into then what we're doing in the lab. We're working in the splice-switching oligonucleotide, and this is just a slide meant to kind of, again, reiterate with splice-switching, you can do a lot of different things to splicing to have different outcomes. So you can skip exons that have termination codons to correct an open reading frame. You can disrupt an open reading frame by skipping an exon and creating a termination codon to decrease gene expression. You can block cryptic splice sites if there's a disease caused by a mutation that creates a cryptic splice site, or you can regulate alternative splicing. We just talked, we heard also from John Prensner, who is doing, thinking about targeting non-canonical ORFs. Something like this could potentially be done through splice or steric blocking mechanisms as well, or even RNase H gapmer. So they really can apply to almost anything. We've applied the splice-switching type oligonucleotides to look at a number of different diseases: cystic fibrosis, Parkinson's, Usher syndrome, Alzheimer's disease targets. But today I want to talk to you about a rare disease called CLN3 Batten disease. So this is Jessica Senta and Matthew Stratton, a postdoc and graduate student in the lab, who've done most of this work that I'll talk to you about today. CLN3 Batten is a rare genetic disorder. It's a lysosomal storage disease. It has autosomal recessive inheritance. It's the most common pediatric neurodegenerative disorder as a whole. The whole Batten disease, CLN3 is a subset of that. Onset is around 4 to 8 years with vision loss. So there's an eye component, and then seizures. Onset typically after that, you get a loss of motor function, gradual, and then more rapid cognitive decline, psychiatric problems, and typically death occurs in the late teens and early 20s. A hallmark is the storage material accumulation in the lysosomes, and there's really no cure or effective treatment. We work a lot with the 4Batten Foundation, the founders of which have 2 girls with the disease, and this is one they're younger, and here they are today. So most of common cause of the disease is the deletion of exons 7 and 8. And most patients have this. 85% of patients have at least 1 copy of this. And this results in a termination codon in exon 9. And so one approach here, these types of large deletions are often very difficult to find approaches for using antisense or really anything. Gene therapy hasn't proven very effective for this particular disease, mostly because we don't know what the function of the protein is, and we don't know a lot about the expression of the protein with no good antibodies. And so that's been a limitation of understanding expression levels and where to express it. So we had the idea that we would correct the open reading frame by skipping exon 5. If you skip exon 5 using an antisense oligonucleotide to block splicing of that exon, you make an isoform. It corrects the open reading frame. So you get a full length protein that's missing. It has an internal deletion. This is a lysosomal membrane protein, typically modeled to have 11 transmembrane domains. Again, with a termination codon, it's a very truncated protein, and we also know it's very unstable. So the RNA is degraded through nonsense-mediated decay. This isoform rescues the stability of the RNA and is predicted to make a protein that would still be integrated into the membrane. This rationale for this approach was bolstered by the fact that there's a lot of spliced isoforms of CLN3. These are the cDNA, the identified isoforms from Ensemble. And we've noted that there was one in particular that has been annotated that is missing exon 5 and exon 7 and 8. And this isoform is also conserved, has been found in macaques and dogs as well. So it's a naturally occurring spliced isoform. And further rationale, in rare cases of this disease, there are people that have mutations in CLN3 and all they have are retinal or vision problems. And so our real hope is that we can take a very severe disease, do open reading frame correction with our antisense oligonucleotides and make a less severe disease, ideally with even minimal visual deficits. So where we started with this was we're fortunate to have, and we then made some mouse models. We engineered, or there was a knockout model that deleted the same exons that the humans had. And then we made a therapeutic model where we knocked out exon 5, as well as exon 7 and 8 to sort of mimic genetically what we wanted to do with our ASOs. And I don't have time to talk about that today. It was recently published. And we showed that we did get therapeutic rescue in mice that only expressed this deleted exon 5, 7, and 8 compared to a mouse that had the mutation, the human mutation. And so that was really good support for the fact that ASOs might have some therapeutic value. So we started screening ASOs. This is a typical in vitro screen to identify good candidates for further screening in vivo. This is just an RT-PCR after transfection of different ASOs in. So you can see here some skipping that occurred. We then, I'm just going to abbreviate this story since we have published this, but we treated mice one time when they were first born. And then we did a series of behavior tests and analyzed splicing and histology at various time points. We were really excited by how great this oligo worked. After neonatal injection out to 14 months, we still had really good, here's the skipping activity. Some of it is lost, but it lasted for a long time. This is an antibody to the ASO. These are untreated, and then the ASO is staining in green. These are the treated ones, and you can see in the hippocampus, the cortex and the thalamus, this is just a blow up. You can see we get really great distribution of the ASO throughout the brain. And this is the RT-PCR of these different brain regions. You can see we get really good skipping here, and here it is quantitated. And so the oligos work phenomenally well. And then just, we did a number of behavioral analyses also, and this is really just one of those. We have a severe model of CLN3. So unfortunately, the human mutation in mice, the mice are not really, they don't have a severe disease. And so that is one problem that we dealt with when trying to show efficacy for our oligo. But we made a severe mouse model where on this genetic background, this severe genetic background, there is a shortened lifespan. So 50% survival is about 115 days. If we knock out one copy of CLN3, lifespan goes down to 59.5 days. So survival is dependent on CLN3 copy number. And then if you have both CLN3 genes having the mutation, you get very severe phenotype with death around 50% survival on 18.5 days. And so we are really excited when we treat these mice with the oligonucleotide, we recover survival to the heterozygote survival. And so this is, to us, a really good validation of this approach as a possible therapeutic. And then we also screened for the human ASOs, targeting the human gene in patient fibroblast cell lines, and found a lot of oligos that do skipping. So here's the full length and the skipped. So we have a lot of really active oligos. We moved into a pig model. So our collaborators at Sanford Research had made a pig model with the mutation, the Delta 7-8 in pigs. And we really like this model for a number of reasons. First of all, we can screen some of the human ASOs in pigs because they're more conserved than mice. And also the possibility of being able to screen for the visual deficits, because the pigs our collaborators at Stanford show that they do start to have some visual deficits. And so this is we're in the middle of this study, and I just sharing the beginning. We're just starting to get some visual, ERG data on these pigs. We give one ASO injection of the animals, and this is our splicing data. And I'll just point out this percent skipping over here, whether we use 300 micrograms, 200 micrograms, or 80 micrograms. Out to 6 months, we're having really good skipping retained. And when we use 200 micrograms out to 12 months after a single injection, we're getting 70% skipping still. So we're really excited about this pig model and hope to share more with you about the visual rescue in the pigs very soon. In my last 1 minute, I want to say, so Matt said in the beginning, I came here to the University of Michigan to help create this mRNA Therapeutics. We really aim and have a vision to be an all-inclusive RNA therapeutics center and help labs within Michigan and outside of Michigan to develop treatments, discover and develop treatments by helping consult and design, synthesize, and work with clinicians and others in the preclinical and clinical development, and really trying to go from the bedside to the bench and bench to bedside by accessing patients, getting genetic information, designing therapeutics, and getting it back into the patient. So we're really excited about doing that. Whoops, I forgot to say Peter Todd is my co-director, clinical director of mRNA Therapeutics, and happy to talk with any of you about it that want to know. And here's, again, my acknowledgements, a lot of collaborators helping with the CLN3 project, and this is the funding that for those projects. Thank you.
Thank you, Michelle. That was great. So yeah, type in questions in the Q&A. I have a quick question first. So it's good to see that FDA sort of regulatory approach to this N of one, N of few, it's changing, right? It's becoming a little bit easier to get approval. What is sort of the off-target effects of ASO that you may worry about? What would be the worst scenario? I mean, it seems like they bind RNA and they do a splicing switch, but what could be sort of some of the bad effects of ASOs?
Yeah, so there's off-target in terms of not hitting, so doing complementary base pairing in a site that it certainly shouldn't interact with. For the gapmers or the RNase H targeting, that can kind of have catastrophic events. So that's, it definitely needs to be carefully screened and assessed. The steric blocking is a little bit higher bar to have an effect, just because you would have to base pair and then also hit a region that was sensitive to, that you are blocking cis regulatory region or something like that. Mm-hmm. But it's something that we also do screen for mm-hmm. in terms of, and that's something you can predict bioinformatically and also, empirically as well.
Okay. Great. Question from Tim Robinson. My experience with ASOs to induce alternative exon usage is that the alternative or novel isoform can be difficult to express or detect at the protein level. What strategies do you use to decrease nonsense-mediated decay and help promote expression of alternative non-native isoforms?
Yeah, that's a good question. I mean, it is a really important thing, because when you do an oligo, especially a splice switching oligo or any type that's targeting RNA, you might predict, okay, it's going to skip the exon, but it might skip 4 exons, or it might activate a cryptic exon. You might not see it because it's lowering it because it's getting degraded. And so we typically do 2 things. One, we'll treat with puromycin or something to block NMD, and see whether or not we then see new isoforms. And also we look at the absolute level of the RNA through qPCR, just to see where we are at in a downstream or common region, to make sure that something isn't happening to the RNA at changing levels that we're not aware of. So those are kind of our 2 approaches.
Okay, one more question here from Thushara. what's the toxicity over the scrambled control?
So we typically don't, this is a really bigger question. It sounds like a simple question, but it's something we talk a lot about. So we don't typically do a scramble control. We do a non-targeting control a lot of times. And that is, again, a good question, right? Because that can have all its own problems, right? Whether it's on target or that. So we typically use the same one for most of ours, and the one that we are using, we've never seen toxicity, but it is something that I think you do need to have the proper controls of your vehicle and your control oligo to make sure that it's not your control oligo that's having an issue.
Okay, one last question here before we have to close. This is from Fabrice again. Any specific hybridization independent OTEs with proteins?
Oh, that's a good question. So yeah, a little less is known about this. I think you're asking about whether it can sequester proteins, which is definitely an area of interest. There are, I mean, that's how oligos get into cells. They bind proteins, circulating proteins, and that's one big way that they can get into cells. And so, and also once it's in the cell, there have been oligos that are designed to actually act as decoys. And so that is something to think about. There's also effects, since some of the oligos, depending on the chemistry, are charged and some are not charged, and that can cause local changes in the ion pH also. So it's a really good question, and it's another thing that has to be considered if you're seeing toxic effects.
Okay, that's all we have time for today. So thanks again, Michelle. Thanks, John, for your talks. And that's all from University of Michigan this time. Thank you. Bye-bye, everyone.
Thank you.